Good afternoon. We are students from Harmony School of Advancement presenting our PBL project, testing for the presence of Neisseria meningitis using ELISA.
The ELISA test, or enzyme-linked immunosorbent assay, is a quantitative and qualitative test that detects the presence and amount of antibodies, antigens, and hormones. This test can be used in three different forms: a direct ELISA test, an indirect ELISA test, and a sandwich ELISA test. In an indirect ELISA test, which is the one we use for our experiment, either the antibody or the antigen can be tested for by depositing the known or unknown antigen, primary and secondary antibodies, enzyme, and substrate into the well. Depending on whether the test is positive or negative, the solution will either turn blue or remain clear. The ELISA test is a scientific wonder, as it can identify unknown antigens and antibodies. It has many practical uses currently, such as COVID tests, pregnancy tests, and more.
After completing the experiment, we found out that patient 1 tested positive for Neisseria meningitis and patient 2 tested negative. While 19 to 21, patient 1 were light blue, matching the positive control, while 13 to 15, while 22 to 24, patient 2 were clear, matching the negative control, while 16 to 18 showed. On top of these results, we also measured and determined the strength of the pathogen infecting the patients by comparing the shade of blue to the standard curve in wells 1 to 12. As patient 2 tested negative for the bacteria, this is only applicable to patient 1 who tested positive. The wells of patient 1 came out as light blue, which means that the sickness is not that strong. To gain more accurate results, 3 trials were ran for each of the variables: positive control, negative control, patient 1, and patient 2. One possible source of an error in our experiment could be that when we were inverting the samples over the sink to determine if the antibody had bound to the antigen, the sample may have been shaken too hard, displacing the antibody even when it has bound to the antigen. As expected with our hypothesis, the patient with Neisseria meningitis came out blue, and the patients that tested negative came out clear. Since patient 1 was confirmed to be positive for this sickness and their samples came out blue, vice versa with patient 2, our hypothesis was supported. The ELISA test is an extremely applicable and useful technique in the real world today. In fact, the ELISA test is at the forefront of ever-growing COVID-19 pandemic, being one of the main ways to test for the presence of the virus.
So how are enzymes, antibodies, and antigens involved with the ELISA test? In the ELISA test, an enzyme is used as a protein that catalysts a biochemical reaction. Additionally, an antibody or antigen immunological molecules may be involved, resulting in a good outcome if the molecules react or a negative outcome if they do not. How can ELISA test detect an antigen in bacterial meningitis? An ELISA test can produce qualitative or quantitative data depending on how much detective chemical is present. Qualitative results indicate whether a patient is positive or negative for the presence of the antigen or antibody in the bacterial meningitis. It is seen that the substrate binds to the enzyme bound to the secondary antibody, causing the color to change. This results in the antigen being present.
So, who contributed to the discovery of the ELISA test? One of the researchers that helped develop ELISA in 1971 was Eva Engvall, who was born in 1940. She was awarded a PhD in 1975 by the University of Stockholm. She completed her postdoctoral study at the City of Hope National Medical Center in California and the University of Helsinki, where she was later hired as staff. She was hired in 1979 by the Lajala, a California-based Sanford Burnham Medical Research Institute. Engvall held dual positions from 1993 to 1996 at Stockholm University's Department of Developmental Biology and Sanford Burnham Medical Research Institute. Thanks for watching our PBL presentation, Harmony School of Advancement.